Hello, welcome you back for the further topic in the nanomaterial and the characterization. In this, now we are discussing is with respect to the optical microscopy. This optical microscopy comes under the topic of the characterization. Now here, the main question is what is the need of the study of the optical microscopy? And as we know that the optical microscopy or the microscope, it is used for the magnification purpose. Optical microscopy is nothing but magnification through the visible light. Now the term characterization is refer to to the material such that the structure and also the properties are probed and measured. Even the characterization, what we are measuring it, either with respect to the dimension or with respect to the shape or with respect to the type of the particle or type of the element what it is there, that it is done either in macro level or micro level or even the nano level. For that reason the optical microscopy is most essential to ascertain the material characteristics. An optical microscope is also sometimes known as a light microscope. Uses one or a series of lens to magnify the images of a small sample with a visible light. The lenses are placed between the samples and the viewer eyes and to magnify the image so that it can be examined to the greater details. The simple microscope, or it may be a compound microscope, or it may be a digital microscope, or even the stereo microscope, or the comparison microscope, or even inverted microscope. These are the different types of microscopes which are available under the optical microscopy. These microscopes normally what we are using to study up to either it may be for the micro level. For that reason only it is called as the micro and scopy. Next, here in this slide what we are discussing is with respect to the microscope. Microscope is the tool which can help you to see a tiny objects, either it may be a living organism or it may be a non-living organism, into a bigger size. The ability of the microscope is for the magnifying or the magnification factor. The magnification factor is represented in the form of X. The microscopes also have a capacity to distinguish between the gaps between the two particles, or it may be the two separate points which human eyes cannot be distinguished. This resolving power and the resolution is also makes the microscopy a more advantageous in identifying the characteristics of the material. Operation and application and limitations. In order to use the optical microscope effectively, it is important to ensure that the microscope is set up correctly. The objective lens should be brought close to the steady sample to allow the light inside the tube of the microscope. This creates an enlarged inverted image of the sample which can be viewed through the eyes of the microscope. Optical microscope is commonly used in many research areas, including either it may be the biology or even electronics or even physics or biotechnology and pharmaceutical researches. There are some instances when optical microscopy is not well suited to the task at the hand due to the limitations of the technique. For examples, at very high magnification, air discs may be visible, which are fuzzy discs surrounded by a diffraction rings which appears in place of the point objects. When the limitations of the optical microscopes are significant, alternative types of microscopes may be more useful. Now, the microscope. The pictorial representation of the microscope shows that a compound light microscope. The term light refers to the method by which the light transmits the image to your eye. The compound deals with the microscope having more than one lens. Microscope is a combination of two words. Micro means small, scope means view. Early microscopes are called as simple because of only one lens. The simple scopes works like magnifying glass that we can see, or it can be used as something like a magnification glass, normally what we are using it. These are early microscopes had limitations to the amount of the magnification no matter how they are constructed. The creation of the compound microscope by the Jensens helped the advance the field of the microbiology light years ahead, and where it had been only just a few years earlier. Jensens added the second lens to magnify the image of the primary image or the primary lens. The schematic diagram shows the compound microscope. Now, the characterization of a small structure or a small size material in the nanoscale usually calls for a sophisticated characterization tools. The characterization of a nanomaterial or a nanostructures has been largely based on the certain critical advancement and conventional characterization method developed for the bulk materials. For examples, XRDs, SEMs, TEMs, and so on. For examples, XRDs has been widely used for determining the crystalline structure, crystalline size, and crystalline material properties and lattice constants, nanoparticles, and nanowires and nanothin films. Scanning electron microscopes and TEM together with an electron diffraction have been commonly used in the characterization of the nanoparticle to get the idea of the size, shape, and defects present in these materials. The optical microscopy is used to determine the size of the semiconductor quantum dots. SPM. The scanning probe microscopy is a relatively a new characterization technique and has found a widespread application in nanotechnology. The two branches of SPM and scanning tunnel microscopy and atomic force microscopy. Although STM, the scanning tunnel microscopy, and atomic force microscopy are the true surface imaging techniques that can produce a topologic image of the surface with an atom resolution in all the three dimensions. In combination with appropriately designed attachments, these characterization technique and their application in nanotechnology level. Next, the characterization and manipulation of the individual nanostructure requires not only the extreme sensitivity and accuracy, but also atomic level resolution is important. For that reason, the characterization and measurement of a nanostructured material is most essential. STM and as well as AFM have found much broader range of applications such as nano uh, indentation and nanolithography and patterned self-assembly. Almost all solid surfaces, whether the hard or soft or electrically conductive or not, can be studied with the STM and as well as AFM. The surfaces can be studied in the gaseous media such as air or vacuum or even the liquid. In this chapter, we are briefly discussing in detail. Next, the first one what we are discussing is with respect to the scanning electron microscope. The schematic diagram of the scanning electron microscope consists of the electron gun, the first condenser lens and second condenser lens. This comes under the category of the compound microscope. Electron beam light is emerging from the gun and the same is reflected or the same is transmitted through the lens, and it is uh, impinged on the sample, by which the magnification it can be achieved. The magnified image can be seen through the computer. The scanning electron microscope is one of the most popular and widely used technique for the characterization of the nanomaterial effectively. In addition to the information of the surface topography, SEM can be used to provide a useful information on the chemistry, crystal orientation and internal stresses distribution. The image magnification can be achieved in the range of approximately a 10 to over 300,000. The SEMs consists of electron gun to emit the electron that are focused into a beam with a very fine spot size, approximately 5 nanometer. The electrons are accelerated by the energy value and the range of a few hundreds of electron volt to 50 kilo electron volts, and rested over the surface of the specimen by the deflection coils. As the electron strikes and penetrates the surface, number of interactions that results in the emission of the electrons and the photons from the samples occurs. The SEM image are produced by collecting the emitted electrons on the cathode ray tube. The various SEM techniques are differentiated on the basis of what subsequently detected and imaged. The principal image produced in the SEM are three types. The first one is secondary electron image, backscattered electron images and element X-ray maps. The disadvantages of the SEM. The main disadvantage the main advantages of the SEM are: SEM includes a wide array of the application in finding out a three-dimensional and topological images and the versatile information gathered for the different detectors. SEMs are also easy to operate with a proper training and advance in the computer technology and associate software to make an operation a user friendly. The main SEM limitations is the size and as well as the cost. And another disadvantages with respect to the SEM is very expensive and large and must be housed in an area free from dirt and even the electric, magnetic and vibration interference. The maintenance involves a very huge cost. Electronic coils and the circulation of the cooling water is also another major disadvantage. The special training requires to operate the SEM as well as the to prepare the samples at the minute level. The preparation of the samples can result in artifacts. Artifacts is nothing but the tooling which is used for the preparation of the samples. The negative impact can be minimized with the knowledge of experience of the researcher being able to identify the artifact from the actual data and as well as the preparation skill. In addition, SEMs are limited to solid or inorganic samples, small enough to fit inside a vacuum chamber that can handle the moderate vacuum pressure.